Neurological disorders, understanding inheritance patterns, biochemical basis, and clinical presentations. In this comprehensive script, we will delve into the intricacies of neurological disorders, exploring their inheritance patterns, biochemical basis, clinical presentations, genetic testing methods, population genetics, management strategies, prenatal testing and counseling, differential diagnosis, neuropathological changes, carrier screening programs, current research advancements, and genotype-phenotype correlations. Through detailed analysis, we aim to provide medical students with a deeper understanding of these complex conditions and equip them with the knowledge necessary to diagnose and manage patients effectively. Let us embark on this enlightening journey into the world of neurological disorders. Neurological disorders exhibit a remarkable diversity in inheritance patterns. Autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and mitochondrial inheritance patterns have all been observed in various conditions. Autosomal dominant inheritance is characterized by the vertical transmission of the disorder from an affected parent to approximately 50% of their offspring. Conversely, autosomal recessive inheritance necessitates the presence of two recessive alleles, resulting in a 25% chance of manifestation for each offspring. X-linked disorders exhibit different inheritance patterns in males and females due to the presence of the X and Y chromosomes. Additionally, mitochondrial disorders are maternally inherited through the cytoplasmic transmission of the mitochondrial DNA. The biochemical basis of neurological disorders often lies in dysfunctions within metabolic pathways, ion channels, neurotransmitter systems, or structural proteins. For example, Huntington's disease is caused by an unstable expansion of CAG repeats within the Huntington gene, leading to the abnormal aggregation of the Huntington protein. Similarly, in amyotrophic lateral sclerosis, ALS, mutations in genes such as SOD1, C9orf72, and TARD disrupt protein homeostasis and impair neuronal function. Deficiencies in enzymes or transporters essential for neurotransmission, such as phenylalanine hydroxylase and phenylketonuria, PKU, can also lead to neurological manifestations. Neurological disorders encompass a wide spectrum of clinical presentations, ranging from subtle cognitive impairments to severe motor dysfunction. Symptoms may include progressive muscle weakness, tremors, cognitive decline, seizures, sensory disturbances, or psychiatric symptoms. The clinical presentation is often influenced by the specific genetic mutation involved, making accurate diagnosis crucial for appropriate management. Advancements in genetic testing techniques have revolutionized the diagnosis of neurological disorders. Molecular techniques such as polymerase chain reaction, PCR, Sanger sequencing, next-generation sequencing, NGS, and chromosomal microarray analysis have widely been employed to identify specific genetic variants associated with neurological conditions. These tests aid in confirming diagnosis, determining carrier status, and providing valuable prognostic information to patients and their families. Tay Sachs disease is a rare neurological disorder primarily affecting individuals of Ashkenazi Jewish descent. It is caused by a mutation in the hexa gene, which codes for an enzyme called beta-hexosaminidase A. This enzyme is responsible for breaking down a substance called GM2 ganglioside in the brain. In individuals with Tay Sachs, a deficiency of this enzyme leads to the accumulation of GM2 ganglioside, causing progressive neurodegeneration. The prevalence of Tay Sachs is higher in Ashkenazi Jewish populations due to a founder effect. This occurs when a population descends from a small group of ancestors who carried the mutated gene. Generally, individuals of Ashkenazi Jewish descent have a higher carrier rate for Tay Sachs compared to the general population. Screening programs have been developed to identify carriers of the Tay Sachs mutation. This information is helpful for family planning decisions and prenatal diagnosis. These programs have significantly reduced the incidence of Tay Sachs in certain populations. Understanding the population genetics of Tay Sachs disease is crucial for public health interventions and genetic counseling. By studying the genetic variation and prevalence of the disease in different populations, we can better allocate resources and develop targeted strategies to prevent and manage Tay Sachs effectively. The management of neurological disorders involves a multidisciplinary approach, encompassing pharmacological interventions, physical and occupational therapies, supportive care, and genetic counseling. Pharmacotherapeutic modalities aim to alleviate symptoms, slow disease progression, and improve quality of life. Personalized medicine, tailored to the genotype and phenotype of individual patients, holds promise for more effective treatments in the future. Prenatal testing is pivotal in identifying high-risk pregnancies and facilitating informed decision-making. Techniques such as chorionic villus sampling, CVS, and amniocentesis along with newer non-invasive prenatal testing, NIPT, methods provide opportunities for early detection of neurological disorders. Genetic counseling, an integral part of prenatal care, assists families in understanding the risks, implications, and available options for management or intervention. The differential diagnosis of neurological disorders involves a comprehensive evaluation of the patient's clinical presentation, family history, and laboratory findings. Distinguishing between overlapping symptoms can be challenging, necessitating careful consideration of various genetic and acquired conditions. A systematic and inclusive approach is crucial to arriving at an accurate diagnosis. Neuropathological changes associated with neurological disorders provide insights into disease mechanisms and aid in confirming diagnoses posthumously. Examination of brain tissue microscopically reveals hallmark features, such as amyloid plaques and neurofibrillary tangles in Alzheimer's disease, Lewy bodies in Parkinson's disease, or demyelination plaques in multiple sclerosis, MS. Carrier screening programs are important tools used to identify individuals who carry recessive genetic mutations that can cause certain inherited diseases. These programs are particularly focused on populations with higher rates of these disorders. By identifying carriers, individuals and couples can make informed decisions about their family planning, reducing the chances of passing on the disease to their children. These programs are beneficial in reducing the burden and providing support to affected families. Research endeavors continue to expand our understanding of neurological disorders, paving the way for novel therapeutic approaches and potential cures. Advancements in gene editing technologies, precision medicine, and stem cell-based therapies offer hope for the development of targeted treatments to ameliorate symptoms or halt disease progression. In the context of Tay Sachs disease, genotype-phenotype correlations have been extensively researched. In most cases, the presence of specific mutations in the hexa gene, which encodes for the enzyme beta-hexosaminidase A, is associated with the classic infantile form of the disease. However, there are rare cases where individuals with the same mutations exhibit later onset or less severe forms of the disease, suggesting the influence of other genetic or environmental factors. Studying these correlations is crucial for accurately predicting disease progression, prognosis, and tailoring personalized management approaches. It also enables genetic counseling to inform families about the potential risks and outcomes associated with different genotypes. Neurological disorders present remarkable complexities in terms of their inheritance patterns, biochemical basis, clinical presentations, and management. Through genetic testing, population genetics studies, and ongoing research advancements, we gain valuable insights into their underlying mechanisms. A comprehensive understanding of these disorders equips healthcare practitioners with the knowledge necessary for accurate diagnosis, effective management, and informed genetic counseling. As the field progresses, continued research and collaboration hold the promise of fighting these debilitating conditions and improving the lives of those affected.